Hi everyone, welcome to SPAK 24. I am Adewumi Adeniyi. I will be teaching you biology, and today we'll be looking at the cell. The cell. The cell has been defined as the building block of all living things. The cells are the building block of all living things. And both in terms of structure and function, the cell is the basic unit of life. We are talking about structure, function. The cell is the basic unit of life. And usually it's made up of protoplasm that is surrounded by cell membrane. Briefly, let's look at the discovery of the cell. The cell was first discovered, or the word or the term cell was first used by the man Robert Hooke in his publication titled Micrographia that he released in 1665. He coined the term cell to describe the structure, the chamber-like structure he discovered when working on a piece of cork at the back of a plant cell. What he worked upon was actually a dead cell in a plant. When he looked at it using a simple microscope, he discovered that the structure was like small chamber. It was a simple microscope he used back then. So working with this simple microscope, he discovered that there was some chamber-like, some compartment, empty spaces. There were spaces without anything inside. Of course, according to the capacity of the camera, the microscope he worked with, so he couldn't discover much inside the cell. Then all we saw was that there was empty spaces, empty chamber, okay, that looked like small room. That was why he coined the term cell to describe the structure he saw. And when you look at the honeycomb structure, when you say the honeycomb structure of cell, it was because this chamber, this room-like chamber, this compartment he saw while working on the piece of cork, they look similar to this small chamber that you see when you are looking at the honeycomb structure. I mean, when you are looking at the structure of the honeycomb of bee, you discover that they look similar to what. That was why we use the word the honeycomb structure of cell. So till today, if you are in any situation and somebody say who discovered the honeycomb structure of cell, it was still the same man by the name of Robert Hooke, because the structure he saw then when he was working on a piece of cork, they look like the honeycomb structure from the bee hive. So that is why he coined it, the people credit, or that is what it today. We credit the honeycomb structure of cell to the work of Robert Hooke. Now let's look at the type of cells. There are two principal types of cell. We have the prokaryotic cell and we have the eukaryotic cell. Of course, when you look at the diagram I put here, this is a bacterium. The cell here, from the word prokaryotic, pro means before, and karyot from karyon. That karyon refers to nucleus when you use the word karyon. So that was the cells, to say that the cells evolved before the evolution of nucleus. These cells evolved before the evolution of nucleus. So we say prokaryon or before. So these cells do not have true nucleus. They don't have true nucleus. Instead, there's just a region. There is a region in the cell. That region is called nucleoid, where you have most of the genetic material of the cell. You call it nucleoid. So like I said, there's no nucleus. So you just have a nucleoid. But then, so the word prokaryotic cell, there's no true nucleus. There is no membrane cover. So most of the organelles that are found in prokaryotic cell, they are not covered by membrane. They are not covered by membrane. But when you talk about eukaryotic cell, the eukaryotic cell, they evolve after the evolution of nucleus. So that means they contain true nucleus. So eukaryotic cell contain true nucleus, and they are organelles. When I use the word organelle, what do I mean? Organelles are any structure that you find inside the cell and that possess a specific function. So any structurally and functionally distinct part of the cell are referred to as organelles. Okay, so organelles inside eukaryotic cell are covered by membrane. They are surrounded by membrane. Unlike prokaryotic cell, where the organelles are not covered by membrane, in eukaryotic cell, the organelles are covered with membrane. An example of prokaryotic cell, of course, we have here bacterium. Okay, bacterium is an example of prokaryotic cell. And when you talk about examples of eukaryotic cell, we have plant cell, we have animal cell, even cells of protozoa, cells of fungi. They are eukaryotic because they have two nucleus and they are covered by membrane. Now let's briefly look at the forms in which living cell exists. The forms in which living cell exist. It was a man by the name Felix Dujardin in 1835 who first recognized the existence of one-cell organism, and he claimed that protoplasm seemed to be associated with all forms of life. So he first recognized the existence of one-cell organism. What do I mean? That is, you have a single cell that is carrying out all the characteristics, all the characteristics of features of life within the framework of a single cell. A single cell can carry out the process of reproduction, nutrition, cellular respiration. They do everything. All the characteristics of living cell are carried out within the framework of a single cell. Okay, and when you have cell like that, you say the cell can exist. That is, the cell exists as a single free living individual. A single free living individual. Okay, so a cell can exist as a single free living individual. Example of such cell, of course, is amoeba, paramecium, chlamydomonas, euglena. They exist as free living individual. They exist as free living individual. Another form in which living cell exists is as a filament. Okay, as a filament. When you have cells connected together end to end, when you have end-to-end connection of cells, as you have in Oregonian and you have in Spirogyra, you have end-to-end connection of cell. So cell also can also exist in form of a filament. And 3, cell can also exist in a colony, as a colony, in a form of colonial organization, where you have cells connected by cytoplasmic strand. Okay, cells joined together by cytoplasmic strand. Of course, here you have many cells together that they depend on one another. But if it's not that in colonial organization, if you take a single cell out, that cell is not going to die. As soon as you take a single cell out of the colony, that cell will quickly undergo the process of cell division to reconstitute a new colony. So it will undergo cell division and form a new colony for itself. Okay. Now, finally, cells can exist as part of a living organism. Cell can exist as part of a living organism. In this case, you have different type of cell. For example, the body of human being, there are millions of cells. We have cardiac cell. We have those are cardiac cells in words of the heart. We have rods and cones in the eye. You have epithelial cell. You have cells in the kidney, billions of cells constitute. In fact, our brain alone made of several billions of cells called neurons. So a cell can exist also as part of a multicellular organism, So briefly, I said cell can exist as a single free living individual, 2 as a filament, 3 as a colony, and 4 as part of a whole multicellular organism. Now, briefly, since we listed all the proteins when we are talking about cell as a free living individual, let's briefly look at comparison between the proteins. Of course, I made mention of about 4, but out of these 4, 2 of them, Euglenan and Chlamydomonas, they are both photosynthetic proteins that show they have some plant life feature, by which I mean they can carry out photosynthesis to manufacture their own food. And amoeba and paramecium, they are animal-like, meaning that they are non-photosynthetic. They are non-photosynthetic proteins. They cannot carry out photosynthesis. Okay, so we have Euglena and Chlamydomonas showing plant feature. They have some plant feature, though those of them, they also can, they have a part that can make them respond to light, something like that, but they can carry out photosynthesis. Then we have paramecium and amoeba, both of which are non-photosynthetic. Now, let's look at a few adaptations of paramecium. Paramecium have some adaptation. They have contractile vacuole, which can help them to eject excess water, because one of the problems confronting them in freshwater habitat is how to get rid of excess water from the system. And they also have cilia, which help them to move from one place to another. They have pellicule that give them structure and shape. Of course, that is why they are not shapeless like their friend, their near friend, protists like amoeba, who does not have a shape. It's shapeless because there is no any support. One of the things you must have learned from a supportation and support system is that support system gives shape and rigidity. So since amoeba does not have a support system, they are shapeless. But in the case of paramecium, they have pellicule. So this pellicule gives them shape. Okay, and inside the pellicule there is what we could there is a thread-like structure called trichocyst. This trichocyst also help in protection. They can shoot out this trichocyst to entangle their prey. It can even make them to look up or to appear bigger. Okay, that is for paramecium. So they have so that the fewer ways by which they have an advancement over amoeba, which is close to them. And when you look at Euglena and Chlamydomonas, since I said that both of them are photosynthetic in nature, they have life-sensitive parts that help them to move to an area where there is light, where life is available. But they store Euglena, for example, store paramecium. Euglena store paramecium, while Chlamydomonas store starch. Now, what's the difference between the two of them? While paramecium is a glucan that consists of a glucose unit. A glucan is a polysaccharide made up of a glucose unit that is linked together by beta-1,3 glycosidic linkage. Okay, I said glucan is a polysaccharide. Both of them store carbohydrate. Both of them store polysaccharides. Okay, both of them have chloroplasts that contain chlorophyll, which can store polysaccharide as their own food reserve. But in the case of Euglena, they store paramecium. It's a polysaccharide that is made up of glucose unit linked together by beta-1,3 glycosidic linkage. While Chlamydomonas store starch, like any other lamp plant, they store starch, which consists of glucose unit linked together by alpha-1,4 glycosidic linkage. Okay, alpha-1,4 glycosidic linkage in the case of straight chain compound, but when you are talking a branch of branch chain compound or starch, they are linked together by alpha-1,6 glycosidic linkage. Now let's briefly look at the cell theory. Like I still have to display the picture of a man by the name Robert Hooke, and this is microscope that he used in his work. This is a microscope. This is a picture of the proteus of the man. Okay, since he was the first person to coin the term cell, so I'm not just going to jump and begin to talk about cell theory, without mention of his name. Now briefly, so there are 3 scientists that make significant contribution to the development of the cell theory. The first is a man by the name Matthias Claudin in 1839. This man worked on different type of cell and came up with a statement that, oh, I have worked on the body of so many plants and I've discovered that all the bodies of plants that I work on so far, they are made up of cell. At about the same time, his German colleague by the name Theodor Schwann came up with a similar statement that in my own case, I work on the body of plants, of animals. I work on the body of animals. And I discovered that all animals are made up of cell. That was the breakthrough that led to the formation of the cell theory. Now said that, okay, that means every living thing, since living things consist of plant and animal. So that means all living things are made up of cells. If plant is made up of cell, or if the body of all plants are made up of cell, and the body of all animals are made up of cell, that means the body of all living things consists of cells. So that was when we have the first statement of the first one. I say the cell is a basic unit of life. Then all living things are made up of cell. And then at about 1855, another man by the name Rudolf Virchow came up with the statement that all cells came from pre-existing cells through the process of cell division. In his own publication, he made use of the word omnicellular, acellular. That say all cell comes from pre-existing. The meaning of the statement is cell come from cell. Omnicellular, acellular. Cells come from cell. So that was where we this statement was, or this discovery led the human race out of the dark as to what the origin of new cells. So from that time, we can now understand that all cells come from pre-existing cell, and that through the process of cell division. So we have the cell theory. Every living things are made up of cell. You see, or stating them in order to say the cell is the basic unit of life. 2, all living things are made up of cell. 3, all cells come from pre-existing cell through the process of cell division. And lastly, today, based on what we have discovered from molecular study, can say that the information for the growth of the cell is contained in the DNA of their nucleic acid. Information for cell growth is contained in the DNA of their nucleic acid. Now, cell structure and their function. Of course, I defined organelles the other time as any structurally and functionally distinct part of a cell. When you look at my chart here, we have a cell where we have a protoplasm and plasma membrane. Then protoplasm is the living part of the cell, and you have a gastric body. Then you have if you say what constitutes the protoplasm, you have the cytoplasm, and the nucleus. And then if you look at the cytoplasm, it's made up of the organelles that are swimming inside the cytosol. And so it's the cytosol, there are no organelles. But when you talk about cytoplasm, you have the cytosol and you have the organelles. And the nucleus, you have the nucleolus and you have the nucleoplasma. Now, let's briefly, these are the structure of the cell. Now, let's talk about some of these organelles that you find inside the cell. We have the cell wall that cover that is usually adjacent to the cell membrane, usually found in a plant cell. Animal cells do not have a cell wall. That's what differentiates between them. That's why easily when you pour too much of water, if animal cell absorb too much of water, they burst. But in the case of a plant cell, they nearly become turgid. They build to gut pressure and become fully stretched. Okay, so the cell wall constitutes additional support. It surrounds the plant cell and usually pressed adjacent to the cell membrane. It allows cell to develop turgor, and like I said, usually when you have a plant cell that does not contain cell wall, we call them protoplasts. Okay, when you have a plant cell without cell wall, you call them protoplasts. Now, the nucleus is the organelle that house the genetic material of the cell. The chromosome are present inside the DNA, and of course, in the chromosome, the chromosome is made up of DNA. Okay, and On the DNA, you have different genes that code for a particular protein. Now, the nucleus regulates gene expression. It coordinates the cellular activities like growth, metabolism, and reproduction. It's the control center of the cell. Just like you have the brain as a control center for human body, the cell is the control center for the cell. Now, the mitochondria, which is usually referred to as the powerhouse of the cell, okay, responsible for generating energy in the form of ATP. Then you have the endoplasmic reticulum, which could be rough, could be smooth. It is rough when it's studied with a ribosome. It is smooth when it lacks a ribosome, and usually functions in protein synthesis. Ribosome are involved in protein biosynthesis. So since they are attached to the rough endoplasmic reticulum, so they function in protein synthesis, folding, and modification. And the smooth ER are involved in lipid synthesis, detoxification, and calcium ion storage. Now, let's look at the Golgi apparatus, which is a multi-compartment organelle that function in protein and lipid modification, sorting and packaging, in fact, and distribution to whether they are going to go out of the cell or stay. Then the lysosome, which is usually referred to as the suicide bag of the cell. When the cell is whole, the lysosome can break down to it contains a lot of digestive enzymes that play roles in apoptosis or programmed cell death. So instead of allowing cell to stay there and continue to generate inflammatory reaction by the cell immune system, the lysosome can play a role in destroying them and making them to undergo apoptosis. Okay. Now, the vacuole, that like large membrane sac that store a lot of ion, nutrient, and waste product. They function in plant cell. They are central. We have a very large centrally placed vacuole that helps the cell to maintain the turgor pressure. It also stores pigments and facilitate growth by absorbing water and regulating the volume of the cell. Then we have the chloroplast, which is the site for photosynthesis, a process whereby green plants manufacture their own food. Okay, of course, the chloroplast contains chlorophyll, which is inside the chlorophyll. You have the structure called the grana. The thylakoid of the grana can trap light energy in the process of photosynthesis. And we have cytoskeleton, which function as the support, okay, for the cell. And then we have the cell membrane that could determine what moving and out of the cell. Then we have this table here talk about the differences between plant and animal cell. Difference between plant and animal cell. Of course, we have usually the plant cell. They have a chloroplast, and there is no chloroplast in animal cell. In plant cell, we have plasmodesmata. There's no plasmodesmata in animal cell. And in animal cell, the nucleus is centrally placed, while in plant cell, the nucleus is at the peripheral region. And usually in animal cell, centrioles are present, but in plant cell, there is no centrioles. Centrioles are present in animal cell, but in plant cell, there is no centriole. Right now, let's talk about properties and function in the cell. Of course, there are many lot of them. We have metabolism, cellular respiration, nutrition, growth, etc. Briefly, what is metabolism? Metabolism is the sum total of biochemical reactions that occur in a living cell. It's divided into anabolism and catabolism. Anabolism has to do with the building up of organic material, while catabolism deals with the breaking down of organic material. Of course, cellular respiration, where food is broken down, is a catabolic reaction, while photosynthesis is an anabolic process. The Krebs, they also talk about cellular respiration, where they say manufacture energy. Of course, the first stage in cellular respiration is glycolysis, where glucose is split into 2 molecules of pyruvic acid. And then after glycolysis, if there is oxygen, the cell will enter into Krebs cycle, where energy is generated. A lot of coenzyme that can store energy that will be used, that can store proton that will be used in the electron transport chain to generate large amount of energy in the process of what we call oxidative phosphorylation, electron transport chain, and chemiosmosis. Okay. Now, briefly, we are going to answer some questions. Let's treat some questions on the cell. The first question I have here says, which of the following controls the metabolic activities of the cell? Which of the following control the metabolic activities of the cell? And the first thing I have here is chloroplast. Chloroplast, and we have a ribosome, we have lysosome, and we have nucleus. Of course, I said in the course of the teaching, when I was talking, I said that the nucleus control the metabolic, because the nucleus contain all the genetic material that are expressed, okay, and the enzyme that are going to form the actually protein, which are made based on the code that has that present the genetic material that present in the nucleus. Okay, when they are transcribed into functional product. So the nucleus control the metabolic activities of the cell, because the nucleus contain the code of the genetic material of the cell. The genetic material is what that are the genetic material are transcribed, the one that are transcribed in the process of protein formation. The genetic material are transcribed and then translated into protein. So it is the nucleus that controls the metabolic activities inside the cell. The next question says, euglena uses flagella for dash: A, respiration; B, fighting; C, protection; D, locomotion. Of course, flagella is a locomotory organelle. So the use of flagella by euglena is to move from one place to another. Other locomotory organelles in the animal kingdom include, we have the legumen, you have the fins of fish, and when you look at the protozoa, you still have flagella used for movement in some, we have a cilia. For example, in amoeba, we have a pseudopodia. Pseudo means force. Okay, in amoeba. All right, so let's look at our next question. The next question says, the function of the contractile vacuole in paramecium is dash: A, to remove excreta; B, for locomotion; C, to produce enzyme; D, to get rid of excess water. The correct answer is to get rid of excess water. Of course, paramecium is a freshwater organism. Paramecium is a freshwater organism. And in the course of evolution, the main problem confronting organisms that live inside freshwater is, how can they get rid of excess water rushing into their body? How can they get rid of excess water rushing into their body? And that was what led to the evolution of contractile vacuole, in which when they, as they move through, of course, you know, the body of the cell is hypertonic to the surrounding liquid. So it's only normal for water to move by endosmosis into the cell. So when water rushes into the body of the cell, the contractile vacuole will be absorbing, the water will be entering into the contractile vacuole. When the contractile vacuole become completely filled with water, it breaks down to discharge the water to the exterior, after which another contractile vacuole will be formed. So the function of contractile vacuole in paramecium is to eject water, to get rid of excess water. The next question says, a chain of reaction through which almost all living cells produce energy. An aerobic reaction is called dash: A, protozoan cycle; B, carbon cycle; C, organism cycle; D, Krebs cycle. Of course, when you look at everything put here, they are just put there to distract you. There's nothing like protozoan cycle. We have carbon cycle, of course, but that's not what you are looking for. Carbon cycle is the process that bring about the continuous circulation of carbon. Of course, organism cycle, no. So Krebs cycle or crassulaceous acid cycle It's a process that takes place inside. Why we talk about glycolysis the other time. Why glycolysis take place inside? Why glycolysis take place inside the cytoplasm? Krebs cycle take place inside the mitochondrion, which is the powerhouse of the cell. So inside the mitochondria, you have the Krebs cycle, where a lot of coenzymes that will be used, like I said earlier, in the electron transport chain will be generated. So the Krebs cycle is a chain of reaction where almost all the living cells produce energy in aerobic respiration. Let's look at our next question. Which of the following is known as the powerhouse of a cell? Which of the following is known as the powerhouse of a cell? Option A, we have ribosome, cytoplasm, nucleus, mitochondria. Of course, ribosome is involved in protein biosynthesis. Cytoplasm is where all activities inside the cell take place, or where most of the organelles inside the cell swim. Nucleus is the core, while the mitochondrion is this organelle where cellular respiration takes place. Mitochondrion is the home of cellular respiration. So inside the mitochondrion, we have cellular respiration taking place. Cellular respiration produces energy in the form of ATP, adenosine triphosphate, and because of this, the mitochondrion is referred to as the powerhouse of the cell. The next question I have here says, which of the cell organelles contain the DNA? A, cell membrane, B, lysosome, C, cell wall, D, nucleus. Which of the cell organelles contain the DNA? Now you look at it, all these organelles here, the cell membrane doesn't contain DNA. It's only made up of a bilayer of lipid, where you have some protein studied. Lysosome is the suicide bag of the cell that contain mainly enzymes. The cell wall, only found in plant cell, is just adjacent to the cell membrane, like we said, and help the cell to be strengthen the cell, having it to be able to become turgid. But looking at the nucleus, it is inside the nucleus of the cell that you have the genetic material of the cell. Inside the nucleus of the cell, you have the genetic material of the cell. So the nucleus is the one that contains the DNA. It contains the DNA of the cell. The only other organelles where you can find DNA which they are not part of your option, of course. So that is only where, if you have them, you might be confused. If you have the mitochondrion and the chloroplast, because those two cells organelles are product of endosymbiosis. Endosymbiosis is a prayer whereby it was believed that the ancestors of today's eukaryotic cell were once swallowed up you have a bigger cell swallowed up by a smaller cell, and the two of them were able to continue living together in a process called endosymbiosis. And some of the evidence is you see that those organelles have two membranes. They have an outside membrane and they have inner one, and both of them have their own DNA. Aside those two, all these sets of organelles you find here, none of them contain DNA except nucleus. The next question says, the cell found in the brain is called, we have chloroplast, leucoplast, nephron, neuron. Chloroplast is present in plant cell and is contain chlorophyll that is able to trap the sunlight energy and convert it to chemical energy stored in the molecules of food that are manufactured. So chloroplast is found only in plant cell. Plant doesn't even have brain. Leucoplast is not found in the brain. Nephron is only found in the functional unit of kidney, where urine formation takes place. And neuron, the neurons are the specialized cells of the nervous tissue, of the nervous system, that stores information and carry messages within the nervous system and between the nervous system and every other body system, every other part of the body. So the neuron is the cell here that is found in the brain. Of all the four, the neuron is the only one that is found in the brain. The neuron is found in the brain. Okay, so even leucoplasts, when you check very well, you discover that even leucoplasts, they are found in plant as well. Like I said, that plant do not have a brain. Okay, so the next question, we have the organelle responsible for removal of water from the body of protozoa is called A. Nucleus, B. Cell wall, C. Chromatin, D. Contractile vacuole. Of course, we've already discussed this before, that contractile vacuole is the organelle that is responsible, that get rid of excess water from the body of protozoa like amoeba and paramecium, and I discussed that it evolved based on the problem confronting the organisms that are living in the freshwater ecosystem. Of course, every organism will have some challenges facing them based on where they live. So the freshwater organism, their own problem is that the body is hypertonic with respect to the surrounding liquid, which means the gradient of concentration will be concentrated and will be created, and down which material will move, okay, across the cell, the selectively permeable membrane, and that is why the water will rush into the body of the animal, and they have to deal with the water. That was what led to the evolution of the cell membrane. Now, which of these cell organelles is not found in animal? Which of these cell organelles? Cell membrane, ribosome, mitochondria, or chloroplast? If there is no cell membrane, there will be trouble. Of course, we said that the man eukaryotic will consist of the body made up of eukaryotic cells, and eukaryotic cells contain membrane. Okay, so membrane, cell membrane is found in animal cell. Ribosome. If there is no ribosome, how will they synthesize proteins? So animal cell contain ribosome, and if they don't have mitochondria, they'll be powerless. Where will they be generating all the energy for numerous metabolic activities that occur in the body of man? So, but when you look at chloroplasts, animals don't have chloroplasts, of course. They don't manufacture their own food. Animals are heterotrophic, so they don't contain a chloroplast. The next question, what is the name given to group of similar cells performing the same function? Of course, we have organelles, we have system, we have organ, we have tissue. A similar cell or a group of similar cells, structurally and functionally similar cells, are referred to as tissue. So the answer is tissue. We then can have tissue plus tissue to form organ. Organ plus organ will form system. And of course, the cell is made up of organelle. Okay? Now, let's look at the next question. I have a question 11. I say, the living materials of the cells consist of dash. A, cell membrane and cell wall. B, vacuole and ribosome. C, Cell 1 and cytoplasm. D, nucleus and cytoplasm. If you look, of course, we have the cytoplasm, where inside the cytoplasm you have all the organelles. The nucleus is at the center for animal cell, though might be squeezed to one side in plant cell, but the nucleus and cytoplasm are that the living materials of this, or the living material consists of the nucleus and the cytoplasm. That's what form the protoplasm of the cell. The protoplasm of the cell. The structure in cell concerned with the synthesis of protein is the dash. A, we have nucleus, we have cell membrane, we have Golgi body, and then we have ribosome. Which of them is concerned with the biosynthesis of protein? The correct answer is ribosome. Fine. Nucleolus inside the nucleus make the ribosome, and as soon as the ribosome is made, it leaves the center, the nucleus, and comes attach itself to the endoplasmic reticulum, making it rough. Or you can find some directly swimming inside the cytoplasm. But usually, ribosome are found attached to the endoplasmic reticulum, making it rough. And the function is to synthesize protein. So the structure in a cell that is concerned with the synthesis of protein is ribosome. When the solute concentration of a cell is higher than the intracellular solute concentration, then it is said to be dash. First, what do you mean by intracellular solute concentration? Intra, not inter. Intra. Okay, when the solute concentration of a cell is higher than the intracellular solute concentration, then the cell is said to be dash. You see, dilute, isotonic, hypotonic, or hypertonic. When the solute concentration of a cell is higher, okay, the cell is said to be hypertonic with respect. Okay, there are three terms here. We have hypotonic, we have hypertonic, and we have isotonic. So they are usually relative. They are relative terms. You use them. You don't isolate a system and say the system is isotonic or hypertonic. There must be another thing you are comparing it with. Okay, so when you have a cell surrounded by a cell membrane, okay, and the solute inside the cell, the solution inside the cell contain more solute. Okay, when they contain more solute than the other solution, you said the one that contain more solute is hypertonic with respect to the surrounding one that is having lesser solute, which is then described as being hypotonic. And of course, when you have the two situations where both of them have equal concentration of solute, like you make a solution contain of 5 ml of water, inside which each of them contain 1 cube of sugar each, and they are separated by a cell membrane. These two solutions, of course, will be said to be isotonic. Iso means equal. That is, the two solutions have equal solute concentration. Okay, so when the solute concentration of a cell is higher than the intracellular solute concentration, the cell is said to be hypertonic. Our next question says, which of the following solutions causes shrinkage of living cells? A, isotonic solution. B, hypotonic solution. C, deionized water. D, hypertonic solution. Of course, hypertonic solution will cause living cells to shrink as water begin to move out of the cell by exosmosis. The cell will be completely plasmolized. Okay, so the cell will shrink. So that process is called plasmolysis. The next question says, which of the following cells is a specialized cell? A, plasmodium. B, meristematic cell. C, amoeba. D, guard cell. When you look at plasmodium cell, they are not. They are just like amoeba. Option C. They are cells that call as a free living individual. carrying out the characteristics of living thing within the framework of a cell, like I said earlier. The meristematic cells, they are cells that are capable of active division. They are not specialized. But when you talk about gas cells, they are already specialized for a particular function. Specialization is a process whereby either you say specialization or differentiation, process whereby the cell becomes specific in action, in function, whereby they become specialized to carrying out a specific function. That is what we call cell specialization. Other examples of cells that are extremely specialized, when you talk about neurons, cells you find in the hair, in the eye, rod and cones. Our next question says, which of the following organism consists of two or more cells? A, we have paramecium, B, we have euglena, C, we have amoeba, and D, we have volvox. Of course, the first theory, we've already discussed them earlier, that they exist singly. It's a free living individual. But for volvox, we have many cells that are connected by cytoplasmic strands. So volvox is the right option there. And the next question says, which of the following organism does not have a definite shape? Which of the following organism does not have a definite shape? Protozoa. We cannot generalize because there are many organisms in the protozoa. Then paramecium. Of course, paramecium has shape because of the presence of a pellicle that imparts shape. We discussed this in the course of the teaching earlier. Euglena too is not shapeless. They have shape. And when you talk about amoeba does not have any support system, and as a result of that, amoeba is shapeless. So the correct answer there is amoeba. The next question says, the organelle used for defense in paramecium is known as, the organelle used for defense in paramecium is known as dash A, endoplasm, B, pellicle, C, cilia, D, trichocyst. The organelle used for defense in paramecium is known as that. Here they are specific. We have endoplasm. That is not, that's out of the way. When you talk of pellicle, we said that the pellicle contains trichocyst. Okay, it is the trichocyst. The cilia is just for locomotion, but the trichocyst that is present inside the pellicle of paramecium is a thread-like material, it's a thread-like organelle that can shoot out a thread-like substance to entangle the predator that attack the organism. So the correct answer will be trichocyst, not pellicle. Because the next question says, the organelle responsible for protein synthesis is called dash. We have cell wall, we have mitochondrion, we have nucleus, we have ribosome. Even though mitochondrion can make its own protein, the correct answer is ribosome, which the main function it carries out is biosynthesis of protein. Of course, the best answer would be ribosome. Okay. The next question says, the two major parts of protoplasm are dash A, cytoplasm and nucleolus, B, cytoplasm and chromosome, C, cytoplasm and ribosome, D, cytoplasm and nucleus. The correct answer will be cytoplasm and nucleus. You can listen to the teaching again. That was mentioned there. Now, the next question says, the mode of nutrition undergoes by amoeba is A, autotrophic, B, saprophytic, C, allophytic, D, holozoic. The mode of nutrition undergoes by amoeba. Of course, amoeba does not. Amoeba they don't have chloroplast, so they cannot manufacture their own food. They depend on the already made food from their environment. So they carry out holozoic nutrition. So they take already made food. Saprophytic cannot work because they are not fungi. They can not feed on dead decaying organic matter and grow there. Then autotrophic will not, because they don't have chlorophyll, and neither is it allophytic. Okay, they are not even plant to start with. So holozoic nutrition, when an organism taking ready-made food from the environment. The next question is, the suicide bag of the cell is called dash. The suicide bag of the cell is called dash. A, mitochondrion. B, chloroplast. C, ribosome. D, lysosome. The suicide bag, of course, mitochondrion is not. We discussed this also in the course of our teaching, that lysosome contain a lot of digestive enzyme, and because of this, they can break. We mentioned the word autophagy. We also talk about apoptosis, programmed cell death. Because of the digestive enzymes that are present inside lysosome, they can break down. They can break down their own cell, break down themselves, the cell. Instead of staying there when the cell is maybe hold or one or the other, and allow the cell to generate inflammatory reaction by the body's immune system, lysosome can break it down. So autolysis is self breakdown. Okay, so that because of the enzymes that can help the cell to undergo autolysis, that is why we call lysosome the suicide bag of the cell. The next question says, the method of reproduction common to paramecium and amoeba is dash. A, transpiration pool. B, budding. C, conjugation. D, binary fission. Binary means two. So the two of them can undergo binary fission and break, where they split into two. It cannot be. Conjugation is a mode of is a form of sexual reproduction, though undergone by paramecium, but not the same with amoeba. Okay, so the mode of reproduction that is common to the two of them is binary fission, a form of asexual reproduction where they split into two. Next question says, the process by which organisms maintain a constant internal environment is called. The process by which organism maintains a constant internal environment is called as A, hydrolysis, B, osmosis, D, dialysis, so C, dialysis, and D, homeostasis. Homeostasis is the maintenance of a constant internal environment regardless of the changes that is taking place in the environment. Homeostasis is the maintenance of a constant internal environment regardless of the changes in the environment. So the correct answer is homeostasis. Question number 25. The final stage in both meiosis and mitosis in a eukaryotic cell is known as dash. A, anaphase. B, interphase. C, metaphase. D, telophase. If you follow PMAT, prophase, metaphase, anaphase, and telophase, the final stage is telophase. After that, we have cytokinesis, which is the division of the cytoplasm. Which of the following mode of nutrition do organisms manufacture their food? A, commensalism. B, symbiosis. C, parasitism. D, chemosynthesis. In commensalism, an organism feeds on crumbs from another organism. In symbiosis, two organisms live together. In parasitism, one organism depends on another organism living in that organism or inside the body of the organism or outside. Okay, so we can talk about endo and ectoparasite. But in chemosynthesis is a form of autotrophic nutrition. You know, when you talk of autotrophic nutrition, auto means self. A form of nutrition whereby an organism is able to manufacture their own food by themselves. Okay, so in chemosynthesis, the energy used for food assembly, for assembling or manufacturing food, come from the oxidation of simple chemical compound. Examples include chemosynthesis in sulfur bacteria, where the oxidation of simple chemical compound like hydrogen sulfide provide chemical reaction that could be used to assemble carbohydrates. The next question says the organelles where oxidative phosphorylation takes place is dash. A, ribosome. C, cytoplasm. B, cytoplasm. C, cell wall. D, mitochondrion. Oxidative phosphorylation takes place in mitochondria. Okay, when you have after Krebs cycle, we have a lot of coenzyme, a lot of nicotinamide and other nucleotides and flavin and other nucleotides that are produced. This will enter into the electron transport chain and generate a lot of proton that will descend back into the matrix of the mitochondrion using the route provided by ATP synthase. And as a result of this, organic phosphate is able, the energy generated there will help organic phosphate to combine with ADP to form adenosine triphosphate. Okay, so oxidative phosphorylation take place inside the mitochondrion. Next question say the process of cell duplication in which one cell divides into two genetically identical daughter cells is called dash. A. Diuresis, B. Duplication, C. Meiosis, D. Mitosis. Of course, diuresis is talking about being diuretic, as found in by when somebody excretes excess amount of water at a time. And duplication is to divide into two. Meiosis is a cell division where cell divide and give rise to 4 daughter cells with haploid number of chromosomes. But in mitosis, the cell divide to give rise to two genetically identical daughter cells. So the correct answer is mitosis. And which of the following cell doesn't contain a cell wall? Plant cell, bacteria, fungi, animal cell. Animal cell does not contain cell wall. Animal cell does not contain cell wall. And the last question I have here say, in the absence of oxygen, dash acid is converted to alcohol in plant. Tartaric acid, citric acid, lactic acid, pyruvic. Of course, pyruvic acid is produced in the process of glycolysis. And this pyruvic acid, in the absence of oxygen, will be converted to alcohol and a molecule of carbon dioxide in green plant. Okay, we call this process alcoholic fermentation. Of course, fermentation is whenever there's complete breaking down of glucose in the absence of oxygen. Okay, so that is the last question we have for today. Thank you for listening. Have a nice time.